Okay, everybody. Unfortunately, I'm the one that separates you from the pizza now, but I'll try to keep it short. So I'm going to tell you a few things about quantitative phase imaging, and I plan to first tell you what that is in case you haven't heard of it or are not too familiar. And I'm going to show you both old and new stuff to kind of show you the progression of this field. First of all, let me acknowledge the contributors to all this work. So this is our group. I just noticed our undergrads are completely obsolete here. There are different names. But here's our grad students, and those highlighted contributed with results directly with slides to this presentation. And as you know, pretty much in all the presentations that you've seen in interdisciplinary research, we have to work with other people from other groups. So I collaborate with people professors from electrical engineering, my own department, like Steve Bopar, Rashid Bashir, but also from other departments. This is my wife in the College of Medicine. We had a strong collaboration from the beginning that continues to this day with Martha Gillette, a neuroscience group. Supriya Prasant is a cancer cell biology group, and we had collaborations with them as well. We also on the clinical applications that I hope I'll have time to show you a few slides on. We collaborate with pathologists. So the Provina Hospital is across the street from Beckman. I don't know if you've noticed it. So it's a local hospital that allows us to get samples very quickly. And for more, when we got more serious about cancer diagnosis, we moved to Chicago. I mean, we collaborate in parallel. Dr. Andre Bala gives us has access to a lot more broader range of samples and so on. We even work with theoretical physicists, and it happens to be your colleague from UCLA, Alex Levine, who helps us once in a while to make sense of our data. Sometimes, most of the time, we have more data than we understand. And sometimes their insights in modeling, especially dynamic phenomena, is great. So with this, this is the outline of my talk. So I'm going to start with background and motivation, then I'm going to introduce spatial light interference microscopy, and hopefully I'll have time to go through these applications on cell growth, intracellular transport, tomography, and cancer diagnosis. So first, okay, so what happens to my presentation? I found an error on my computer a minute ago, but I kind of ignored it. Sorry about this. You know what? Okay, let's go back to the same slide. So I'll start kind of abruptly. Usually I have a few slides, but I'm sorry about this introduction. That's missing. Basically what we want to do is to quantify phase shifts at each point in the field of view. And this was one of the first instruments that we built back when I was a postdoc. I've been working on this kind of imaging for a while now. And the idea with this setup is that whenever you try to measure phase shifts, the main obstacle is actually noise. If you take two beams and you separate them and you put them back together, like in a microsun interferometer, or which is this kind of a cross interferometer, right, or a Mach-Zehnder interferometer that goes like this, you always, if you look at the fringes, they're doing this, and it drives you nuts. If you try to fix it, there is no way, unless you figure out a way to put the two beams very, very close to one another. So this setup that was published in 2006, a long time ago, is actually very, very simple. You take a laser, you couple it into an existing microscope, an inverted microscope, that basically gives you an image plane right here. And we use a telescope that's kind of, you can ignore this part here. Basically, this is the image plane relayed at another plane with some magnification. But the idea is that here we put the grating. So at the image plane, you put a diffraction grating that splits the light in different beams, different diffraction orders. And you see what we managed to do is to have a very tight interferometer, right? So this is the first beam, this is the second beam, through the same lens, and then the second lens makes the image back. So you have an image that goes through this, that is carried by this beam, and the second diffraction order that comes at an angle. So this gives you very, very stable fringes just because they go through the same set of optics. And in order to have one beam that carries the image and the other one into a reference, we filter the second beam through a very small pinhole. So this length creates here for Fourier transform, and basically, it's wow, everything is going well. How are the batteries?
I have one. You want to try it?
Sure. Thank you. If you have my introduction slides, that would be great too.
I can share mine that I haven't used.
Yeah, so I can blame it. You know, we already had introduction on phase measurement, so there we go.
Yeah, from the side there's a switch.
There, boom. Because of these two beams being very, very stable, we were able to actually measure this kind of precisely these kind of phase shifts that come from, in this case, red blood cell. You look at the red blood cell that is fluctuating, and when we first measured this, I remember I took the first movie, and then I post-processed it, and I looked at it, and I realized the membrane is doing this. I thought I discovered a completely new phenomenon. I was so excited, and then we found papers back from 1890s about this. This actually has a name. It's called flickering for obvious reason. But I think we were probably the ones that could measure it the best back when we measured this. So we were able to do it very fast, so 100 frames a second. And this is just because the interferometer was very, very stable that you are able to measure this kind of fluctuations. These are the RMS fluctuations on a red blood cell. On a normal red blood cell is about 30 nanometers or so. Yeah. Papers on the flickering, yes. 1890s. So they were measuring of course not in phase. They were looking at just bright field, and in intensity it's flickering. This is kind of giving you some kind of interference. So hence the name flickering, and that's. No, you look at them. Yeah. So actually, if you look at the regular bright field, I encourage you to do this. Poke your finger. Do the safety first. You actually see them. If you focus on one, you see a little bit of flickering, I guess. Anyway, I think we took this a little bit to the next level because we quantify these fluctuations in the membrane, and then it becomes a very interesting physics problem, and that is kind of if you measure the spectrum both spatially and temporally from a membrane, are you able to back up the physical properties of that membrane? It's very similar to, when we listen to drums, right? From the sound that you hear, you can kind of infer which one is the stiff one, right? Which one is the low frequencies from down here. You can imagine that's a big one, right? As opposed to I forget their names here. This is the bass, this is the principal. I don't know. But it's kind of the same idea. So this is where we collaborated with Alex Levine, who actually wrote a serious model to back this up. So from our fluctuations with certain analysis to fit to his model, we're able to back up shear moduli and bending moduli of the membrane, which is kind of, for the first time, done in a non-contact mode. So people have tried to do this with AFM levels. As you can imagine, that it is doable, but it's a poor measurement, and if you try to cover the whole cell, it's messy. So we published a number of red blood cells. We were very happy, but the reality was, when you're trying to look at something like this, like a neuron that Professor Salvin just showed us, our laser-based imaging looks like that. And if you compare it with the phase contrast image from the 30s, it was kind of putting us to shame. And the reason for that is, and I think Dr. Ozkan mentioned this, is that basically the laser light is too coherent. It gives this random interference pattern, which is called speckle, that basically washes out the details, the internal structures of your object. So that's when I left MIT as a postdoc. That's when I finished my postdoc. And then I moved to here in Beckman. And the number one priority for us was to fix this problem. So how can we do all these quantitative measurements on dynamic samples, but actually with speckle-free? So how can we do this? And I think we fixed this. We call it spatial light interference microscopy. And basically, this is how it works. We maintain the same idea of using an existing microscope. And I like to kind of remind students why this is important. Because probably all of you, when you start to build a setup, you buy stuff from Thor Labs and make your own microscope, like you should as an optics fanatic. We did the same thing. And then you realize after a while, if you want to do any serious biology, all these accessories that come on the microscope, all the stability that comes with the Zeiss or Nikon base are kind of useful. So whenever we can, we keep the base intact. And notice right now the difference is that we don't even change the illumination. So this is a halogen lamp, white light lamp, which normally doesn't go in the same sentence with interferometry and phase measurements, but basically this is the same setup that gives you phase contrast. So back from the 30s, when Zernike invented this, he used white light. This phase contrast, right, predates the invention of lasers by 2 decades. So how many of you know how the phase contrast microscope works? 2 people, excluding faculty. Okay, well, let me try to kind of hand wave it to you in 30 seconds. The idea is to understand that any image is an interference process, and the interference happens between the light that goes through the object. Let's say my object is here. The light that passes through undeviated interferes with the light that scatters. That's the most effective way to think of your imaging. Any image is an interference process. And this was actually written down by Abe back in 1870s. So Zernike understood this idea, so then he treated an image that lacks contrast with an interferogram that lacks contrast. So how do we maximize the interference contrast between 2 beams in general, in Michelson, in any interferometer? What's one condition? Something to do with polarization, right? You want the polarization to be the same, okay? That's the same. The polarization doesn't quite change going through transparent stuff. What else? Same power, right? You want the 2 interference beams to have the same power. That gives you the perfect contrast. But then there is one more thing. If you have an interference between 2 beams, you get the interferogram looks like an intensity of 1 plus the intensity of 2 plus 2 times square root blah, 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 cosine of the phase of the difference between the 2, right? So in imaging cells like this phase shift is not that big. So you're looking at the cosine that around the origin looks like that, right? It's a lazy function around the origin. If you expand the cosine in a Taylor series, you get cosine of x is about 1 minus x squared, right? So it's a quadratic effect. As opposed to sine, which is at the origin, the sine is a line. So it's a lot more sensitive. So Zernike did this, changed the cosine into a sine, and got the Nobel Prize for it. And how do you do that optically? We're talking now, going back to our beams that interfere, is this light that goes straight, interfering with all the light that scatters. How do I separate them? Well, I separate them in the Fourier plane. So this is the back focal plane of the objectives. This is where they're separated. The directed light is focused here and here. It happens that the illumination is over a ring. If you only had the plane wave, it will be focused in one point. But if it's a ring, it's focused over an entire ring. But basically, if you put a ring on top of this, and you put here a delay, a layer that delays the directed light by pi over 2 versus the rest, you create phase contrast. Not only that, they managed to do back then to make this phase shifter layer from an absorbing material as well. So that helps absorb the directed light, which is much stronger for transparent stuff, right? Most of the light goes through. So if you manage to absorb it a lot, and it happens to be about a factor of 5 or 6 in intensity, and phase shifted to pi over 2, you go from a very low contrast to something that has very high contrast like this. This is a phase contrast image. So This was such a great technique that it earned a Nobel Prize in Physics for Zernike in 1953. And it's pretty much all the biology labs have a phase contrast microscope that for quickly looking at the cells and just by eye looking at their variability and so on. And the power is in the fact that you don't need to add fluorophores, contrast agents. It's just an optics thing. So that's why we love it, right, as optics guys. So what we did was to take this one more step further, and we thought of the following. What would be if we could control the phase of the ring? So not only have it fixed at pi over 2, but actually to be able to switch it pi, 3 pi over 2, and so on. In other words, change that cosine into a sine, minus cosine, minus sine. You would be able to go around the whole circle, the trigonometric circle, and you'll be able to get the phase difference accurately and uniquely. So what we did was to project this plane right here onto a spatial light modulator, which is this. So you see these are conjugate planes. This is a liquid crystal spatial light modulator, this device that is able to control the phase at each point in a certain plane. Okay, some of these projectors have these devices in them. Actually, they have 3 of them for each color. So if there is no modulation at this plane, the light reflects off, goes through this second lens, through the reflected off the beam splitter, goes through here, and you get an image that is exactly the same as what the microscope gives you, meaning you get a phase contrast image. But if I'm adding now a pi over 2 phase shift on top of the previous one, then I get another image. If I add another pi over 2, I get another image, and you notice they're all different. And another pi over 2, another image. I combine these 4 through a very simple kind of operation, and you end up with this. So notice, compare this with this neuron that you get with a laser, and basically you notice the improvement. So we see all these now details. The speckles are gone. In fact, they're not gone. What happens is that you get the speckle pattern for each color. Right, so you have a color where you get one speckle pattern that looks as ugly as the previous one. You put another color, another one. When you put them all together, they average out. That's kind of the idea. Okay, so we wanted first to check to see how accurate this thing is. So we took this is a film that is it's a structure like this that has a 4 nanometer kind of height. So it's like a grating almost, and from Professor Rogers, so we took our image with SLIM and then the image with an atomic force microscope, the corner of that thing, and then we compared these two. So you see, this is a histogram that shows the green, the valleys of this structure is the first peak, and then the top, the blue, are the second peak. So you see, they agree within a fraction of a nanometer, and of course, imaging with AFM takes a long time, so it took them to image this corner about 20 minutes. It took us half a second to get the whole thing. The drawback is that in AFM you actually get super resolution. You are not diffraction limited, of course. We are still in X and Y, we're diffraction limited. But in terms of topography, the accuracy is actually very good, sub-nanometer. It turns out that the noise, spatial noise, is of that order. It's a perfect Gaussian of about 0.3 nanometer noise. And if you compare this with the laser background, basically, there is no comparison. This is at least an order of magnitude worse. What's interesting and what was surprising to us is that there is another kind of noise in phase imaging, and that is temporal noise. So if you measure over and over and over, right, if nothing moves, you should get the perfect straight line, but in fact you get noise. Turns out that the standard deviation of that, you see the histogram of that is actually 10 times narrower. So it's 0.03 nanometer path length. And this is because this is the most common path interferometer you can have. You interfere the light that goes through the cell with the light that's scattered a little bit. It doesn't get any more common path than that. So, yeah.
So, like previous slide. So obviously this is a great improvement over the case when you have this cycle force. I'm just wondering, what is this 0.303 nanometer? Where does it come from?
So this is measuring, spatially measuring a flat surface. Path length, optical path length.
But you said this is common path, almost, right? There's no really mismatch between the path.
Let's be careful. You cannot make it perfectly common path because then you measure 0.
right? But if you measure the interference between this light and that light, the phase is locked in place, but is not zero. There is a phase difference between this one and that one if the light goes through the object.
Basically, you're saying if you do this experiment back, where there is no fluctuation, then this should go to zero?
Yeah, and then your electronics is perfect noise and all this stuff. Yes, it will be perfect. It will the only phase difference you will measure is due to your sample. So this noise tells you this, that if I'm imaging something like a cell, right, and the cell somehow swelled up or something, displaced by 0.03, I will be able to see it. That's the ultimate limit. Especially if you give me a layer of something, a film, and the step is 0.3 nanometers, I will be able to see it. That will be my ultimate limit that I can detect in space. As a side note, because you're using an existing microscope, all the other channels are overlaid without you doing anything. You don't need to do numerical localization. So you have here 2 channels of fluorescence of DNA microtubules, and in red channel you see the phase. So for example here, this is an axon. You had the discussion earlier with Professor Salvin, right? Axons and dendrites. Dendrites stain with the microtubulin, with the tubulin stain tag, but the axon doesn't. And what we found kind of as a side note is that the axon, if you look in the phase channel, the axon is smoother. And it makes perfect sense. So if you take a profile of this axon in terms of phase, you get the red curve. If you take the profiles of all these dendrites, you get much more fluctuations. And the reason is exactly the synapses that you saw earlier. So the axon is an input to a dendrite, and it creates these synapses. And at that point, there is a bump. There's probably different concentration that gives you a refractive index. But clearly, there is a physical thickness that is different. So that's why dendrites has a lot of inputs on its shaft, if you want, but the axon has none. The axon itself is an input. So that's what explains. And I'm told that this could be the only way to tell them apart without labeling them. Otherwise, you can never tell which one is the axon. This is kind of more or less obvious, but there are situations where you cannot tell the axon unless you put fluorescence on them. Talking about using existing microscopes and going deeper into biology measurements, I wanted to mention this, that the microscope, oh, you've seen it, right? Some of you in the lab. It looks kind of like this. And if you're trying to build this from scratch, it's a little bit challenging. We did some of it. I mean, my first microscope was a beaten up microscope from Ali Javan's lab, by the way, which was very exciting for me. Ali Javan is the inventor of Hini laser, in case you don't know. But then we needed an incubator, right? And we built our own, some kind of chamber with some feedback. It was a mess. So now we kind of live in a life of luxury, like you have perfusion chamber. Of course, this plastic box costs $20,000. Don't get me started, but everything kind of now works nicely now. You have CO2, humidity control, everything. So long story short, we can measure stuff like this now. You're looking at a movie over 5 days of osteocytes. So this is kind of bone cancer cells. And what I'm trying to show you here is that, first of all, the cells are okay over many days. First of all the accessories work, so the environment, everything. But also the exposure. If you try to do fluorescence over 5 days, you probably kill them, I mean, in the first day. This is a tile, in case you wonder if we broke any laws of physics based kind of bandwidth product. This is a tile of 5 by 5. But also something that was surprising us in a good way is that the nanometer stability is preserved over 5 days. If you asked me this back when I was a postdoc, I would have said this is impossible. But just because it's this common path idea, this is actually doable. So you can image thousands of cells, or you can zoom in and look at high resolution at single cells like this. So again, there was nothing added to these cells. It's all optics, right? So you look at the chromosomes right here. This cell is kind of happily dividing. And this is not even the highest resolution we have. This is a 40x.65 objective. So basically, we kind of know what the measurement is doing, and we're pursuing in our lab. We have been working on a number of techniques from membrane dynamics that I showed you on red blood cells. We're doing blood testing. we're trying to quantify tissue refractive index. So I want you to think of something. What scatters in the tissue? How many of you can answer that question? When you send this light to the tissue, what is scatters? When you get this angular distribution from your tissue, what's scattering? What are you looking at? What structures? So if nucleus? Why? So if you look at the literature, you'll see some people say, oh, we're looking at mitochondria. Some other people say, oh, we're seeing for sure nuclei. Some other people now, more recently, we're looking at stroma. It's almost a matter of taste.
Have you asked this question to Woody?
Yeah, we discussed a little bit. But the reason is that this is a hard problem. This is a mess. I mean, trying to measure this. But what we try to do is to actually now look at biopsies, thin slices of tissue, and it turns out that the answer to this question cannot be black and white nuclei. I think it has to be within, from this angular range to this angular range is going to be this. From this angular range to this angular range could be that. Anyway, I don't have time to talk about this too much. I'm going to show you some biopsies in the end. But try to think of these very, very basic questions. It's actually, they're not very clearly understood. So it's refractometry and scattering, they're basically related. I'm going to show you diagnosis, cell tomography, intracellular transport, and cell growth, unless you're starving to death. First of all, why do we want to measure cell growth? Well, it has been called one of the last big things unsolved in biology. And it's interesting, this is a science paper from Harvard in '09. So they're mentioning this. And they say, obviously, the best method that could possibly do this will be optical. And they cite one of our papers. And then, but somebody has to work on it, or something like that, like get moving. So that was kind of interesting. So this kind of actually triggered us to go back to it, because I'm going to show you how it works, basically. But why is it this unknown? Why do we know not so many things about how cells grow? For example, we don't know whether on average the cells grow linearly in time, mass versus time, whether it's a line or it's an exponential. I mean, think about this. In physics, this will be like before Newton kind of question, right? It's very basic. How are we going to fix cancer if we don't know whether the mass is going? So, but there are reasons, again, why simple things like this are not known. First of all, they're very light. The cells weigh about a couple of hundred picograms, something like that. And another thing from an engineering point of view, there isn't a lot of dynamic range to work with. They double their mass and then drop to 1 again. 2 and 1. There is not 3 orders of magnitude to work with, right? Morphology messes up things. They're not spheres, they're not cubes, they're mushy and deformable. So it's kind of hard. So traditionally people said, you know, forget about the density. We use a cell counter, like a Beckman counter, same Beckman as the Institute, as you know. So basically this gives you kind of a volume measurement. And if you say the density doesn't change, volume and mass is the same, obviously that's not true. But there is no other way. Phase contrast and fluorescence has been used for simple things like E. coli. So maybe there you can say simple structures like that, forget the density, they're just sausages. Okay, fine. But really, the field was kind of, there's a great solution from MIT that basically took this seriously. And they had a very nice idea of having vibrating cantilever, pretty much like in AFM. So you have a resonant frequency of this cantilever, but now you kind of have a channel through it. And when you flow the cells through, the mass of the cantilever changes a little bit, and therefore the resonant frequency changes. So if you measure the shift in frequency accurately, you're able to back out the mass. So I think it's very interesting technology. But now think about this. The cell, first of all, has to be 1 by 1, and they cannot get stuck. They have to continuously circulate. If you want to measure the same cell, you have to make some kind of a loop here, or I think more recently they showed you can have some kind of tube flowing back and forth. But I think the main limitation is the fact that the cells cannot be adherent. And most of the cells would like to adhere. They're happy when they attach to the surface, unless you have red blood cells or some circulating cells. So our own colleagues here, Professor Bashir, tried to fix this problem by now having a platform that resonates. So these are pedestals that have the same idea with the resonant frequency. And now the cells can grow on and divide and so on. So you can look at 1, 2, and 4, and so on. So that's nice. But you also give away sensitivity. Now this is a platform that vibrates in water. There is a lot of damping. In other words, your lines get fatter. And therefore, accordingly, the accuracy in the shift frequency goes down. So with these limitations, we started to go back to our interferometric measurement. And we think that quantitative phase imaging is actually very close to being ideal because you don't add anything to the cells. You can measure one cell, a million cells, adherent or not. And you can decouple morphology. But you may wonder, what is phase imaging to do with mass? And actually, this is the paper that science paper was citing. The concept is very old, comes from the 50s. Comes from the time phase contrast became more commonly used from England. And it's the idea of dry mass. So the dry mass of a cell, and why is it related to the phase? Let me illustrate it like this. Imagine you have a cell of refractive index n on average, and there is some water coming out of the cell, right? So water is expelled from the cell through some osmotic stress, let's say. The phase is actually the product between the refractive index and thickness. So if the water comes out of the cell, two things happen immediately. One, the phase tends to go down because the thickness goes down, right? But there is another effect. The refractive index is more concentrated, right? So the water came out, so you're left with something denser. So basically in this paper we did the calculation where we showed that if you integrate the entire phase profile, the phase surface, you end up with the second order effect in the volume of water exchanged. So think about what that means. It means that if I have small volumes of water coming in and out of the cell, the integral actually doesn't care much. So this we're able to, I think, prove mathematically the idea of the dry mass. That's why this is called, the phase map is proposed the dry mass. It's what's in the cell that's not water, right? It's proteins, lipids mainly. So basically this becomes a direct measurement of surface density of mass, of dry mass. And long story short, we're able to measure things like this with SLIM. So you're looking at an E. coli cell that divides. Obviously, they are on a substrate, so they don't have to circulate. And you can actually track single cells, daughter cells, and so on. And we kind of use the E. coli cells as pretty much the way we use beads as a test sample, because people kind of agree that they have an exponential growth from fluorescence measurements. So we measured a bunch of these. We, from an optical point of view, when you do the conversion to mass, because SLIM is so low noise, you would be able to measure 1.9 femtograms change. That's incredibly small. That's in theory. When you add the culture medium and all these debris in the culture, if you image a fixed cell, which is this one, you see that the noise is actually an order of magnitude higher. 20 femtograms change is still very good to measure pretty much anything. So this is our noise. So E. coli gave us kind of a confirmed that we knew what we were doing. So if you plot, the way you look at the mass change is to plot d mass at dt versus mass, right? So the mass change versus mass. If you get a line, it means that you see d mass over mass gives you dt. So you get mass as a function of time, it gives you an exponential. So this is a good way to check if it's exponential. So people agree that E. coli should grow exponentially. So this confirms that. But we really wanted to look at more complicated cells like this, and this is where we worked with Supriya Prasant, whose name should be here. And they also gave us a good use for our fluorescence channel. They gave us a marker that tells you where the S phase of the cell cycle is. And if you know the S phase, you can figure out pretty much all the phases. So the S phase is where this fluorophore becomes granulated. So what we did, we looked at the fluorescence channels and then at the SLIM channels, so we did both. So basically what we were able to do, so these are the two measurements, we were able to draw curves for each phase of the cell cycle. So just pairing the phase measurement with the fluorescence measurement, so we got curves like this.
So we're able to tell that actually the growth, at least in this particular cell type, which is the osteosarcoma cells, they don't grow equally throughout the phase cycle. It's actually one phase that it seems to be growing the most in this particular cell. We don't know if that's universal, but the reality is nobody knows. I think this is the first time that mass versus time was measured for each phase of the cell cycle. We think this is a good method to test drugs, for example, for cancer. And this is our first paper on kind of looking at the effects of estrogen and estrogen inhibitors on breast cancer. And again, it's very surprising to me that the current state of the art for testing drugs is to get the population number. So you look at from the same dish, from the whole dish, you get one number by kind of monitoring the changing color of a certain chemical in the solution. Okay, so that's the state of the art of testing drugs for cancer right now. And as you know, all cells are different. There is a variability, like with people pretty much. They respond differently. So we think that it's much better to actually, yes, we need to measure thousands of cells, but at the same time have the single cell kind of information. So here you see trends in mass at the individual cell level. We're able to tell that actually when you add estrogen, yes, they grow much faster. There's a lot of growth, but it's not so much through individual masses going bigger, but it's actually through accelerated cell cycle. The cell cycle gets shorter, so they double faster, but in fact, the mass per cell gets down. So you end up with many smaller cells. So that was something that I think wasn't very well understood. So I think this could help kind of understanding the effects of cancer drugs at the single cell level while keeping the statistics for the whole population. I want to tell you a little bit about a related thing, and that is when we work so hard to get these high resolution speckle-free images, and then when you look at cell growth, you throw it all away. You get one number for the whole cell, and you don't use any spatial information. So the original idea was, well, if I take a movie like this, so by the way, these are microglia cells, so these are nervous system cells, in a glia culture. So remember how many neurons in our brain? 100 million. Glia cells are 10 times more, but they don't get that much respect because people thought that they're glue. That's from their name. Their name comes from Greek. But it turns out that now people measured some signaling in the glia, so I think we'll see something interesting. Anyway, the point of this is our original idea was if I have a movie of X, Y, and time, and now these are maps of density, mass density, where it's more red, is denser, shouldn't I be able to understand the physics of the transport? Right? It's kind of an inverse problem. Shouldn't I be able to, for example, back out diffusion coefficients, right? Or velocities? And the answer is yes. And first of all, I wanted to show you here that it may look boring here, lack of signal, but it's just because our RGB map cannot keep up with the signals. Our signal to noise is about 1,000. So this is sub-nanometer, right? And we can measure up to a wavelength, let's say, or more. So if you adjust the colors, you see a lot of stuff here. Something interesting, if you take one gradient along one direction, you end up with an image like this that looks exactly like DIC, which is this kind of a sister method to phase contrast microscopy. And if you tell one biologist that you're doing DIC with a phase contrast microscope, they get really confused. But the reality is that the DIC microscope does this derivative in hardware, right, using Wollaston prisms and whatever. So you see the details pop out. Any derivative is a high-pass filter, right? And what we found, and it has this artifact, right? It has very bright edges. When you go on a bright, high slope positive is bright, and down is negative, is dark. The biologists call it the shadow artifact, and they don't worry about it. They live with it. If you take a second-order derivative, a Laplacian of this image, you end up with this. So you see the artifacts are gone. The equivalent of those artifacts will be positive curvature versus negative curvature. So they are less pronounced. You see even higher details. Look at this. Look at this stuff. You cannot make it up here, but they exist. Our signal, there's a lot of signal in these images, is what I'm trying to tell you. So we are seeing, I think, actin filaments here. So how do we analyze this? So there's a lot of signal. This mass density is measured very, very accurately. You have a lot of dynamic range. But what do you do with it? And I have to say that it's always that good ideas take a while. So we started with something less, I think, less smart first. But basically, the best thing to do is to treat it as any dynamic system where you calculate what is called the dispersion relation, right? Like in optics, for example, the simplest dispersion relation is what? The wave number k equals omega over c. So you equate the spatial frequency k with the temporal frequency omega, and in vacuum, where refractive index is constant, k is linear in omega. Very simple. We did the same thing. So basically, you want to relate the spatial frequencies to the temporal frequencies. So if you take a spatial Fourier transform of this frame, you get a map like this. And we have a bunch of these, right, in time. So then, at each point here, we calculated the power spectrum in time. So now, because we have a distribution of frequencies, we plot the bandwidth of that. So you calculate the bandwidth at each point in qx, qy, in the spatial frequency, and you calculate the bandwidth omega. And if you plot it, you get something like this. And doesn't look very excited, does it, right? It looks like a dish. But now let's plot this profile. So this is the origin. Let's plot omega versus q. If you do that, this is the curve that is pretty amazing. It's pretty universal for all the cells that we ever measured. It has a linear, this is a log-log plot, it has a linear component and then it has a quadratic component. And we model this, again, we worked with Alex Levine. If you model this with the diffusion equation and an advection term, meaning a drift term, the dispersion relation for that is exactly this, a linear term, this is the velocity, and then a diffusion term, which is quadratic. And we can get weird terms like this, q cubed, if you look at filaments, for example. So at even higher q. So basically, what it says is that at high q, diffusion is dominant. So you look at this mass, how it's basically rearranging in space and time, right? So what this curve says that the cell at very short scales, 2 pi over this gives you micron. So let's say below a micron, so 6 will be a micron right here. Below a micron, you have diffusion. Over short scales, diffusion comes for free, and the cell is using that to actually mix things up. Okay, so as we know, for example, at very short scales, like across membranes of the cell, diffusion is the dominant process. So stuff goes inside and out of the cell over the membrane that's only a few nanometers. It's all diffusion. But at large scale, if the cell needs to send a vesicle from the nucleus to, I don't know, in a neuron, right, down the dendrite, diffusion will be too slow. You know the story, right? If we were to respond by diffusion to feel my toe from here, it will take 150 years or something like that. So that's diffusion is just too slow for large distances. So the cell is actually optimizing this thing, where at short scales, it's kind of using diffusion because it counts for free. And then at large scales, it switches to active transport, where the molecular motors that we've just seen in Dr. Selvin's take over. What happens when the cell dies? Well, when we all die, we go to thermal equilibrium. We go to Brownian motion. So the q square goes all the way through all the scales, right? So this part is the region that tells you that the cell is alive. And what's amazing is that this curve is universal. If you look at all kinds of cells, we looked at many of them, this point may go up and down. For example, in a neuron along the dendrite, it's a one-dimensional transport. This thing goes all the way deterministic. So the neurons don't have time for any nonsense. But it's pretty universal. And I don't know, I'm very happy because it's not every day that we're able to put the cell or a living thing in equations. So recently we looked at, try to understand whether we can monitor neuron differentiation and understand kind of this transition from something that looks nothing like a neuron to a fully developed neuron, and then try to understand network formation. And this is a collaboration with the University of Georgia. So we wanted to see how cells grow, how the intracellular transport, this method, maybe how the morphology is developing. So on day 0, you start with this. They look nothing like neurons, right? These are neuroprogenitor cells, embryonic stem cells. Day 3, what you notice is that the sharper structures start to form, but still nothing like neurons, right? They're closer to HeLa cells than anything else. Day 5, now you see the filaments that will become axons and dendrites start to form, right? Day 11, they're very, very close. So basically, we image these over 2 weeks. And eventually, you end up with a network that is fully formed. These are fully mature neurons that basically happen before our eyes. And for one thing, there are not many methods that allow you to image a thing over 2 weeks without killing them, right? So you cannot do this with fluorescence, for example. So that's one thing. But basically, what we wanted to understand is quantitatively, how can we look at these circuits, at these mature neurons? So as usual, first we start by staring at our movies. And if you stare at this as long as we did, you notice something. These cells are extending these kind of actin filaments, bundles, and at some point they make a connection. It's kind of a random search, but then they snap into place, and they leave that alone and they move their attention to some other parts of the cell, and so on and so on. So first we said, okay, how is this network changing mass? And here's what we measured. We're looking from 0 to 24 hours. So how is this network? You put the neurons, they are fully formed, fully developed, put them on a dish. They have nothing to do with one another, obviously, they are independent. You put them in a dish. Number one, how do they grow? How much mass do they produce? Well, here they warm up, they don't do anything, probably they're just attaching and kind of. But then they grow for about 10 hours. So this is significant growth of the whole thing. But then they stop. They grow and then they flatten out. And we did a lithium chloride treatment that kind of prevents the growth, and you get almost a flat line. So there is the growth goes down. So right here, this kind of matches what we observe by just staring at the movies, and that is that they grow enough to make connections with the neighbors, and they don't need to grow. It's as if they have to turn their attention to actually communicating rather than growing. There is no need to grow anymore. So the second analysis that we did was to look at our dispersion relation. So gamma and omega are the same thing. So again, we look at the temporal frequencies versus spatial frequencies. And let's look at the untreated cells. Forget the control for now. So untreated cell at t equals 0, you get the solid line. Look at this. Solid line blue. It's almost perfectly Q squared. Okay? And these are larger scales. So these are kind of intracellular transport, if you want. It's completely random. There is nothing in there. It's all diffusive. But at the end of the 24 hours, if you look at the dashed line, it's almost perfectly parallel to Q. So you start with a dish where the transport is random, and at the end of the 24 hours, it becomes organized and deterministic. So that matches what we were guessing from the beginning. You can calculate velocities, and you see how the deterministic transport velocity goes up. You can measure diffusion coefficients. Finally, we looked at morphology itself. So you start with each snapshot looks something like this, right? So since this is a mass, it's a quantitative map of something, of density, you can do very engineering things, like, for example, calculate correlation area, right, or correlation length, spatial correlation. Or you can look at the power spectrum, the same thing. So we looked at the power spectrum, and here's what happened. At t equals 0, you get the blue line. So this is spatial frequencies. Blue is 0 hours, the red is 24 hours. And what you notice is that high Q, high spatial frequencies, below 15 microns, which is below the cell size, nothing happens. Meaning that the cells, individual cells, stay the same. Nothing happens in their internal structure. Whatever difference you see the trend going down happens at larger distances, smaller Q. This is intercellular distances. All the change happens in how they talk to one another, which again matches the other measurement that we did. And it changes in a very kind of solid trend, right, from blue to red, in the sense that the spectrum is getting narrower, which means the correlation function gets broader, right? At the end of the 24 hour, they are a lot more correlated in space than they started with at t equals 0. So we also show that all this dispersion stuff can be done in fluorescence. So for those of you who are doing fluorescence, in a way, it's kind of generalizing fluorescence correlation spectroscopy, FCS, if some of you are familiar with that, where you actually look at fluctuations from a single fluorescence spot to now look at full field, where it's as if you have a variable spot, from a very small one to a very big one. I don't think we're going to get to cancer, are we? But let me tell you about tomography. This is something kind of recent. So basically, what I'm going to tell you is that you can turn your microscope into a very powerful scattering instrument. And the scattering problem is kind of like this. If I have a cell or an object here, I illuminate it with a plane wave. I'm measuring the scattering wave somewhere far away. And there are two kinds of problems. Kind of undergraduate type of homework problem is we give you the incident light, we give you the object, meaning the refractive index in all 3D, calculate the scatter field. This is the direct problem. That's the easy problem. The harder, more real world problem is we are after the structure of the object, so calculate chi, the scattering potential or the refractive index of this 3D object. You can do whatever you want with your incident field, prepare it the way you want. Be as smart as possible about the scattered field, measure that as well as you want. But the problem is now inverting that and obtaining the 3D structure. So that's a much harder problem. This is the exact problem that has been solved in X-ray crystallography for a long, long time. And this is an incomplete list of Nobel Prizes that stem for solving the inverse scattering problem in X-rays. So starting with Röntgen and Van Laue, who wrote the models for it, the Braggs, all the way to Perutz, hemoglobin structure, DNA structure, all these things are based on this problem of having X-ray incident on a crystal, look at the diffraction problem, diffraction pattern, back out the structure of the crystal. There is a beautiful review by Emil Wolf of X-rays and X-ray diffractions, and the fact that in X-rays you don't have access to the phase at all. They don't have lenses like us. They don't have even detectors. It's a mess. It's very hard to measure X-rays. So if you don't have the phase of the scatter field, the problem becomes non-unique usually. You have to, the structure that you infer might not be unique. You have to come up with some assumptions. And here's what Wolf had to say about this, that not all the guesses have been successful. This is clear, for example, from the following. 2 different structures were predicted for the mineral by this mineral, one by Pauling and the other one by Zaharison. It is not known which, if either, is correct. But in optics, we have these luxuries. Measuring phases now is not a problem. We kind of figured that one out. And of course, the first one to put that on the table was Gabor. A way to, in a funny way, he was trying to fix electron micrographs, if you remember, aberrations in electron micrographs by reading them with light. It never caught on as a electron microscope thing, but it became a field of optics. And he got the Nobel Prize also in 1971. And a paper by Wolf in 1969 took the idea of X-ray diffraction with holography and wrote down the equation that allows you to invert the problem, solve the inverse problem. And this becomes a classical paper that is known as the diffraction tomography paper. And long story short, basically, if you start with an inhomogeneous Helmholtz equation, you end up with the scatter field that is actually, you see in the Fourier domain, is related to the scattering potential. This is just refractive index minus some background. And this is a kind of very simple relationship. You can take chi from here, from this equation, with no problem. Okay? This was done for monochromatic light, single frequency, and scattered field, meaning fields detected far away from the object. The problem with us, SLIM, we wanted to solve the inverse scattering problem, but we needed to generalize the 1969 papers in 2 aspects. One was we had wide light. So this equation doesn't hold for wide light, so we had to kind of integrate over all frequencies. And second, our measurement is in the image plane. So he gave us a lot of a few nightmares for a while. But finally, with Teu, who's your organizer, and the picnic and the grill master, and Renji, who's right there, together they did a great job. And finally, we know how to solve this. So this is the equation that kind of explains our imaging. And the idea is that now, if you take your object and you scan it through focus of the objective, as opposed to scanning angles, so you just scan the cells, focus, and you take phase images at each section. This is the equation that allows you to get chi, the structure, refractive index structure. And I know it looks like abstract and ugly, but let me explain what it means. This is the measurement. Let's say this is the correlation function. This is the phase image, if you want. And basically, in the z direction, you get your structure that is convolved with this mess, meaning that you don't get perfect resolution in z, you get the blurring in z, as we might expect. The whole struggle was, what is that blurring? What is that function? And now we know what it is. And we knew all along that there are two components to it. One is the fact that the coherence effect, the fact that the white light has a very broad bandwidth, and the coherence length that's only about 1.2 microns. But we also knew that the higher numerical aperture, the better. And it's something to do with what Professor Oskan showed earlier, that at larger angles, the scattered and non-scattered fields run out of coherence more quickly than if they're parallel. If they're parallel, they will be coherent all along. If they're at an angle, they kind of go, the correlation goes to zero. So this is the numerical aperture, that aspect, and this is the power spectrum. The broader, the better. The higher the numerical aperture, the better. So in a way, it's sharing the idea of OCT, for those of you who are familiar, of this coherence gating. But it's also very different from OCT in the sense that it doesn't work at zero numerical aperture, for example. While OCT, you can have zero numerical aperture, you still get depth. And numerical aperture and power spectrum are actually very kind of perfectly overlapped. So this is kind of a hard interferometer to build with 1 micron coherent function, but because of that common path situation, everything works. Okay, if you want to plot this in the frequency domain, this is how it looks like. So kx versus kz. So basically, the resolution in x will be how fat this function is. So the width delta kx here, you want that as fat as possible. That will give you good resolution in. This will be in x, and this will be in z. So obviously, kx is something to do with the NA, with the angle, right? So the larger kx, the fatter the kz gets. So that makes sense. We measured small beads and compared our measurement with the calculation. So they kind of match. You see here, they're close. They're not perfect, but it's pretty good. If you add some deconvolution, so once you know this function, you can deconvolve your data with that calculation function. And you see that your PSF is going narrower, so you improve the resolution a little bit. So basically, in X and Y, we're at about just below 300 nanometers. In Z, we're below a micron. We image, for example, these are hard objects to measure. These are single red blood cells that have this crenation. They grow bumps. It's a sign that the cells are not very happy. It could be a transient thing where they come back to a discocyte, or it could be a sign of a bad disease like liver disorders and stuff like that. And you see that if you look at the reconstruction, it looks kind of like the SCM. We are able to look at to section through an E. coli. So this is also hard to do. They are only between 1 and 2 microns thick. And if you look inside, you start to see these helices. This is kind of, people study this for a living. In fact, one of our former colleagues from the physics department here challenged us to see, he's a specialist in E. coli, and he said, we're interested in these helical structures, but we can only study them with fluorescence. Do you think you can see this with your label free? So the answer now is yes. It's not easy, but we can start to see this. You see it here. It's kind of interesting. You have these periodic patterns in E. coli. This is how a eukaryotic cell looks like. This is HT29 cells, so colon cancer cells. And basically, starts to look like a confocal image, except that there is no fluorescence. The cells are happy and alive. You don't need to fix them. And let me show you what we're working on now. So these are HeLa cells, same thing. And what we want to do in the end is to do 3D n times and we understand the transport in kind of all 3 dimensions, do that dispersion thing. And I'm showing this not because it's so great, but because it's kind of ugly and it's telling us that we should be a little bit more in the business, not like Mohammed suggested. So this is look at the cell from the side. So you cut through, this is the glass right here, and you look at the cell. So you see there is stuff here that is not clearly kind of resolved. You look at the funny thing, this is actually a standard deviation in time. Anyway, so ideally we would like to cut through the cells in all 3 dimensions and get views and dynamically at the same time, which is work in progress. Okay, so let me show you one slide on cancer diagnosis. I'm going to skip through everything. This is how biopsies look with H&E staining, which is the traditional way of staining. This is a slim image of the same thing. And we recover pretty much all the details from the stained one in our tissues. You look at the stroma here. These are refractive index bumps due to nuclei, for example. You see them as red bumps here. The organization of stroma is very clear in our images as well. This is a cancer. These are all prostate samples. So you see these glands, very well resolved in the slim images. And this is a bad case of cancer where the glands disappear. This is a very bad kind of advanced cancer. But what I'm trying to tell you is that we recover all the information from the stains into the slim tissue. We've been working on this for a long time. Shamira can tell you a lot more about it. And we think that this will allow us one day to actually have diagnosis on slim, but not only that, maybe predictions of outcomes and things like that, bypassing the staining altogether. So the quantitative phase imaging can give you fluctuations out of plane of thickness, which is what you saw in the red blood cell movies. Can give you in-plan mass transport, and that's kind of the dispersion relation that I showed you. And I talked about these applications. The important thing to remember is that this is label free, so you can monitor them without killing the cells for a long, long time. Thank you very much for your attention. We're ready for pizza. Any questions? Yep.
You still have the characteristic halo of phase contrast. And I wonder if that's ever been a problem.
Yes. Let me show you one halo. So halo is an artifact pretty much like shadow artifact in DIC that people know and are kind of okay with it. That's the way it is. It appears in the phase contrast. You see these bright edges around the cell because of scattered light ending up in the ring where the unscattered light is supposed to fall. And of course, when it goes back to, this happens at edges where you have very strong scattering, right? So that's why. So the light going through that ring goes back at the edge and interferes now with high contrast with the unscattered light. So you get this halo. In our case, you notice this halo is actually diminished. And it's because we're kind of combining 4 frames and some of it is disappears. But clearly, not all of it disappeared. And I think this can be fixed, first of all, if you make this thinner, this will go away. But the reality is we didn't put a lot of effort. We are now working more seriously on this and actually halos in general due to spatial incoherence in DPM and other methods. Actually, you get this halo due to spatial coherence of the light. So we have a good understanding of what it is and how to fix it. Usually, it's not a huge factor unless you really want to be very, very precise when you calculate masses. And in that case, you have to do some processing to eliminate that, because this gives you low values and it may give you an underestimated mass. So that is an effect that we're in the process of solving, but it's there, yeah.
So using this technique, we have phase result images, and then to correlate that back to structural information, we need to know the difference in the refractive indexes as the light is propagating.
No, you don't have to.
the model that I showed you is actually solving for chi, which is n squared minus 1, roughly speaking, or minus the environmental refractive index, without any prior assumptions. It comes out from the calculation like that. So you don't need to assume that.
Able to use this to measure the refractive index of individual components inside the cell.
In principle, yes. But you have to add this other dimension in the measurement, right? Another scanning through Z. From one image, you are correct. The thickness and refractive index comes in a product. But once you solve through Z, essentially you kind of decouple the Z out of the refractive index, kind of.
Because then that's giving you structural information through your scan.
Yes. Yeah.
So there, the Z is not known anymore. You know it's known.
It's kind of like that. Yeah. Other questions? Well, if not, let's have some.